Hello everyone, welcome to my presentation. I am Jayarathna from Department of Animal Science. First of all, I would like to ask question from you. Can essential oils be used as an alternative for antibiotic growth promoters in improving growth performance and meat quality in broilers? As you all know, world population is increasing. As an animal protein source, demand for the broiler meat is also increasing, and this graph shows how the broiler production is increasing to fulfill the demand. However, most of the farm broiler birds are under high stocking density, which cause to rapid spread of diseases and need better strategies to reduce the threat on health and subsequently enhance the growth. Then scientists came up with antibiotic growth promoters. What is an antibiotic growth promoter? Antibiotics that are added to the feed of food animals in order to enhance their growth rate and production performance. But this kind of antibiotics, when we continuously use of this kind of antibiotics, there is a risk of transmission and proliferation of resistant bacteria through the food chain. Therefore, European Union since 2006 and Animal Feed Advisory Committee in Sri Lanka since 2018 banned the use of antibiotic growth promoters in animal feed. Then scientists came up with lots of alternative, and one of the main alternative was phytogenic compounds. What are the phytogenic compounds? Phytogenic compounds are plant derived natural bioactive compound. It has positive effect on animal growth and health and often applied to the essential oil and botanical and herbal extracts. Why phytogenic compounds can substitute AGP? Because it is residue free and generally recognized as safe compound, and it is plant derived natural compound, and it has unique properties like antimicrobial, anti-inflammation, antioxidant and antioxidant. For the phytogenic compound to be an alternative, it should be exhibit the same beneficial effect of more than antibiotic growth promoters. However, there were inconsistencies among the various phytogenic products in literature, and this literature shows gaps in previous finding about phytogenic products. According to that, there were unknown product composition and variable in inclusion levels, inconsistent effect on growth, and lesser studies on meat quality. Therefore, therefore, before the uh, before commercialization of any of these kind of phytogenic products should be scientifically evaluated. Accordingly, objective was to compare the essential one, that is my commercial phytogenic products, and it is mainly consist of mixture of essential oil and plant extract and aromatic substances with the azithromycin 200 WSP, that is my commercial antibiotic growth promoter. It is mainly consist of chlorotetracycline, commonly called as CTC, on the growth performance, gizzard pH, meat quality, and sensory attributes of broiler meat. Material and methods. Study location was Poultry Research Unit and Meat Processing Laboratory in Department of Animal Science. For the growth performance, when 120 after receiving of 120 one-day old broiler chicks weighed and assigned into 20 kg of battery brooders. And here is my treatment plan. All treatments were given with drinking water. For the negative control, only fresh water was used. For the positive control, azithromycin 200 WSP was used, and essential one was given in 2 inclusion concentration. High concentration was 0.1% volume by volume, and low concentration was 0.05 volume by volume. Weekly body weight and feed intake was measured. For the gizzard pH, after 35 days, 2 birds from each cage were taken and euthanized. After that, digestive tract was dissected out. Then 3 pH reading from proximal, middle and distal section was taken. For the Water holding capacity, 2 grams of meat sample was taken, then it was wrapped in filter paper and centrifuged at 2600 rpm for 4 minutes. Then it was vacuum oven dried at 70 degree overnight. Then oven dried weight was taken. For the cooking loss, 100 grams of meat sample was taken. Then it was cooked for 72 degree internal temperature. After it reached 72 degree internal temperature, it was uh, cooked another 30 minutes. Then cooked weight was taken. For the evaluating of lipid oxidation in broiler breast meat, 2 grams of meat sample was taken. Then trichloroacetic 5 ml of trichloroacetic acid was added and then water. After that, 5 ml of thiobarbiturate acid was added and again Vortex. Then it was kept in centrifuge at the 3000 rpm for 10 minutes. After that, it was placed in water bath for 45 minutes. Then it cooled under ice bath. Then absorbance value at 532 nanometer by using spectrophotometer. Then malondialdehyde concentration was taken from the standard curve. Statistical analysis for the growth, performance and meat quality, completely randomized design was used. Mean comparison was done by using least significant difference test at 0.05 significance level. For the growth, performance and meat quality data analysis, SAS software was used. For the sensory analysis, Friedman test and Wilcoxon sign rank test was used, and SPSS software was used for data analysis. And results for the growth performance, weight gain, feed intake and feed conversion ratio was measured. In here, this graph indicates average weight gain of each treatment throughout the experimental period. There was a significant lowest value for the essential one low concentration ES1 treated group. However, there was no significant difference between essential one high concentration and CTC, that is antibiotic growth promoter treated groups. In the case of feed intake and feed conversion ratio, there was no significant difference among the treatment. However, numerically lower feed intake and feed conversion ratio can be observed in essential one high concentration treated group, ES2 group. Gizzard pH is the most important parameter because lower pH improves the pepsin activity and nitrogen retention and increase the solubility of the mineral fraction of the feed. As you can see here, there is no significant difference among the treatment. However, numerically lower pH can be observed in the essential one treated groups. For the meat quality, water holding capacity, cooking loss and lipid oxidation were determined. There was no significant difference for the cooking loss. Essential one low concentration ES1 treated group show the lowest water holding capacity. However, there was no significant difference between essential one high concentration treated group ES2 and CTC antibiotic growth promoter antibiotic treated group. Malondialdehyde is one of the end product of fatty acid oxidation. The lowest malondialdehyde concentration malondialdehyde was observed in essential one high concentration ES2 treated group. It may be due to inherent antioxidant property that present in phytogenic compounds. In the experimental period, a strong aroma was observed in the essential one phytogenic product. Therefore, sensory evaluation for aromatic juiciness, tenderness, taste and overall acceptance was conducted for broiler meat. Then interestingly, there was no significant difference among the treatment for those sensory attributes. I asked question at the beginning. Here is the answer. For the growth performance, answer is yes. We can substitute antibiotic growth promoters for essential one high concentration because there was no significant difference with the antibiotic growth promoter. For the meat quality, my answer is also yes, because there is a no significant difference for the there is a significant difference for the lipid oxidation, and it is very low concentration was observed in the essential 1 high concentration treated group. Therefore, essential 1 0.1% volume by volume can be substituted for AGP. Here is my recommendation. This study was conducted in battery cages in experimental setup. Birds had minimal contact with their feces. However, most commercial farm broilers are reared in deep litter system with stressful condition. Therefore, before commercialization of essential 1 phytogenic product should be evaluated under commercial setup. Thank you very much.